Thank you for inviting me. Today I'm going to be talking about the Model Organism Screening Center that we started in the context of the Undiagnosed Disease Network at Baylor College of Medicine, and this is an effort of 3 teams. Shinya Yamamoto and Michael Wengler are part of this team. So there are 2 approaches that we use to study human disease. The first one is a typical reverse genetics, what we call reverse genetics. We start with human genes and human variants and then create the reagents in the fruit fly to study the mutations in the fruit fly and get a better understanding of the disease. And I'll be talking in the first portion of the talk about this. And then we also use forward genetic approaches to identify genes that cause neurodegenerative disease and then try to link them to human disease through gene matcher and build our cases there. So this is a highly collaborative effort between first human geneticists, obviously, in the reverse genetic approach. And then I'll show you how we work with fruit flies and what we've accomplished in the last 3 years. And the first thing we need to do is really extensive bioinformatic analyses, and we've been focusing on that a lot because we learned very rapidly that when we started working on genes that were thought to be good candidates, they dropped out very quickly. So we need to really do an extensive search in the literature, and we focus mostly on model organisms and the data in model organisms to achieve that. We work with a zebrafish team at the University of Oregon, Monty Westerfield, and John Postlewhite, and so when there is no fly homologue, we turn to the zebrafish. We also work with the comp at Baylor College of Medicine to do the mouse work, and in general what we do when we have an interesting gene is we ask for a knockout if there is no knockout yet made, and then we also ask to make the variants in the mouse so that we can later on continue to study these variants in mice. We collaborate with a set of consortia, so we're obviously part of the Undiagnosed Disease Network, but we also work with the Canadian Rare Disease Foundations. We work with the Center for Mendelian Genomics at Baylor College of Medicine and at Johns Hopkins, and so we're trying to interact with many of these different sites. The pipeline is that, as you know, that there's quite a few undiagnosed disease patients, and they go to these diagnostic odysseys, and then they can apply at Harvard to the coordinating center of the Undiagnosed Disease Network. Upon sequencing the genomes and selecting the variants and the genes that they want to pursue, they can contact the cores. Obviously, the sequencing core, also at Baylor College of Medicine, is sequencing, doing either a whole sequencing genome or whole exome sequencing or whole genome sequencing. There is a metabolomics core, and then there is a model organism screening core. And in the first 3 years, we got about 250 submissions of genes from the human geneticist. And as I said, we go through this bioinformatics pipeline. We developed this program called Marvel. And I'll spend a little bit of time on this because this has really helped us a lot in selecting good variants. And in the first 3 years, we contributed to the diagnosis of about 20 cases. So as I said, Marvel is important, and what we're trying to do with Marvel now is expand Marvel. Marvel queries a lot of the human databases, and one of the main issues we have is that there are so many databases. I think currently we have to query 35 to 37 databases. And you need almost to be an expert for many of them to really be able to mine the data. And so what we do is we have a bioinformatics team that goes through all these data, and what they're trying to do is to present a simple interpretation of what's available in all these data. And the original Marvel, when we came out, essentially queried all the model organism databases, as well as about 7 human databases, but we're now trying to expand that to all these databases. And they're listed here. That is the prediction programs, the protein structure and mapping the mutations to protein structure, the protein-protein interaction databases, and we're also developing artificial intelligence based on all these databases to do variant prioritization. Now, as I mentioned, we collaborate with these other teams, and we also collaborate with the comp, and this is really important because we can then follow up with the mouse knockouts and go back to the teams that submitted the genes and ask if they're interested and willing to work on the mouse later on after we've done our work with zebrafish and Drosophila. Now, the general approach is outlined here, and I want to spend a few minutes on this slide. The strategy that we use in the fly is to insert using CRISPR, a cassette that allows us then to bring in artificial exons into the gene. And so the way we do this is, as is shown in this slide, we insert this cassette here using CRISPR into an intron of the gene, and this cassette can be then exchanged to put in another exon, for example, splice acceptor GFP splice donor, which very often properly tags the protein and allows us to determine where the protein is localized, or we can tag it with a construct that is T2A-gal4. And what this T2A-gal4 construct does is essentially kills the gene, so we have a mutation, but at the same time the gal4 is expressed in the proper temporal and spatial expression pattern. And we can now use the gal4 to drive a UAS cDNA, either a fly cDNA or a human cDNA, and we call that humanization of fruit flies, because in about 70% of the cases, the human cDNAs will rescue the null mutations in the fruit fly, and therefore we can test the variants that have been submitted. And we can do this at a relatively high throughput mode. So using this technology, we can determine with a GFP or an RFP where the protein is expressed. We can drive the human cDNA, we can drive the variants, and we can assess if the variants affect the function of the protein. And we are supported by the NIH to create these reagents genome-wide, so we are supported to create these human US cDNA libraries by ORIP and by NIGMS to create these reagents in fruit flies. So this is just what happened in the last few years with the submissions that we got, and it's just summarizing what we found. First, with the humanization strategy, these are the genes that we identified. We use other strategies. When this strategy doesn't work, we can use a variety of other strategies. And this is just a summary of what all these different strategies are. We can either overexpress a human cDNA and see if it's the wild type is toxic and the mutant is not, or we can do a rescue with a genomic fragment and rescue the mutant in the fly with a genomic piece of DNA and put the human variant in the fly DNA. So we have a variety of approaches. Now, I'll just focus on two cases very briefly. One is ACOX1 and is shown here, and the other is ANKL2 to show you and give you an idea about what it is that we can do. So the ACOX1 story, just a few slides, started with a patient that was a de novo mutation in a 12-year-old kid. He's still alive, but he's now quadriplegic and is not in good health. So the disease was progressive, and when they proposed this mutation in ACOX1, it was kind of weird to me because when we looked at the loss of function of ACOX1, it's a very different phenotype. The diagnosis is typically made within the first few months. The kids have a very short lifespan. They have very high levels of very long-chain fatty acids because ACOX1 is the first enzyme in the beta oxidations of the very long-chain fatty acids. And they typically die by the age of 2 or 3. But this kid was diagnosed at 12, was perfectly healthy till then. And obviously, this suggested that maybe this could be a gain-of-function mutation and very different from the loss-of-function mutation. Through gene matcher, we made a connection with a patient with the same identical mutation in Korea. And that girl was diagnosed when she was 9 and also is now quadriplegic. Now, as I said, ACOX-1 is the first enzyme in the beta oxidation in the peroxisomes. It produces a very high levels of reactive oxygen species. And when it's absent, you have an elevation of the very long-chain fatty acids, as you would expect. But as I said, in these kits, there was no elevation of very long-chain fatty acids. And so we modeled first the loss of function in fly, and then the gain of function, which we thought was a gain of function. And what we found was two very different pathways. The first one, if you look at the variant from the patient, we clearly showed that this was a gain of function mutation, a hyperactive enzyme that created a lot of reactive oxygen species, and that feeding an antioxidant fully suppressed the phenotypes that we observed. And this leads to glial cell degeneration because it turns out we found that peroxisomes are very active in Schwann cells and oligodendrocytes, and therefore the main phenotypes are restricted to those two cells, first the Schwann cells and then the oligos. In the deficiency or the loss of function, you elevate the very long-chain fatty acids and get a very different disease because those very long-chain fatty acids get incorporated in the membranes of the cells. And these cells become recognized as foreign through the immune system, and there is a strong T-cell immune-initiated reaction, and that leads to an autoimmune disease, and therefore the treatment for that is very different, but in both cases you end up with an axonal degeneration. So another example is the forward genetic approach, where we start by inducing mutations in Drosophila, map them, and then through gene matcher try to connect with physicians. And so one of the screens that we did a number of years ago was the screen where we screened for two phenotypes that are neurological phenotypes in the fruit fly. And the first one was to screen for bristle loss using clones. So we induced lethal mutations and then made clones in the thorax and screened for bristles. And this would mean that there is a loss of the peripheral nervous system. And then we also screen in the eye by making clones in the eye with electroretinograms to identify mutations where the electroretinograms would be fine when the fly is closed, but progressively lose the amplitude and the on-offs of the electroretinogram, suggesting that there is a neurodegenerative phenotype. And by doing that, and doing it systematically, and sequencing all these fly strains and rescuing them all, we were able to identify 165 genes. About 33% of those were already, the human homologs were already annotated in the OMIM database, and therefore are shown in yellow here. We were clearly on the right path. Most of these are neurological disorders, but not all of them. So we focused on one gene because we submitted this to Gene Matcher, and through Jim Lupsky and Dobbins at University of Washington got a hit with this ANKL2. And what is shown here was that this kid has a very severe microcephaly. This microcephaly in this kid is one of the most severe microcephalies I've seen so far. And this kid had about 30% of the normal brain volume, and it lived for about 7 years. It died actually only recently. And as I said, this mutation was first identified by a bristle phenotype, and when we saw that this kid had a microcephaly, we looked at the fruit flies and noted that the fruit flies also had a microcephaly and that they had small brains. And these flies have a pretty dramatic loss of neuroblasts. And when we analyzed these, we were struck by the fact that these kids look very similar to Zika patients. In fact, mutations in ANKL2 mimic the Zika virus infections that are occurring in the first trimester in women in South America, to the point that some physicians drew a parallel in the literature before we discovered that the Zika virus targets this protein, and said that there were 3 human genes with microcephaly that should be pursued because they look so similar to what happened in Zika virus infections. And it turns out that the Zika virus encodes 10 proteins, one of which, NS4A, was shown by Nevin Krogan at UCSF to bind to ANKL2 in a large IP mass spec screen. And so he contacted us and asked us to model this NS4A because out of all the proteins that the Zika virus proteins interacts with, there's only one protein that was known to cause a microcephaly, and that was this ankle-2. And indeed, when we express NS4A in the flies, we get a small brain, and we can fully rescue that by overexpressing the human ankle-2. And we've now shown that indeed there is a tight interaction between this NS4A and the ankle-2 protein in that it inhibits the whole pathway. And it's interesting because 3 genes downstream of ankle-2 also cause microcephaly. And so this is a new human microcephaly pathway. And therefore, we think this is interesting because we're going from an N of 1 and a family and a single individual to understanding the molecular mechanisms and the gene-environment interactions, and we're going from an N of 1 to an N of many thousands. There's many thousand cases of microcephaly in South America. So I want to end and come back here to the screen, and in the last 4 years, this table has been filled up, and more than 50% of the genes that we originally identified in the screen are now, through the work of many of you, known to be the cause of human disease. This slide, I want to end with a few more slides showing that this all is based on a very interactive set of strategies between different groups. And the other groups in the world that are part of the Undiagnosed Disease Network International have taken a different approach. So we're a central group and we go through a high throughput strategy, but the Canadian organization, as well as some of the Europeans, have taken a more distributive approach where when they have a gene match between a model organism and a human geneticist, they refer to the person or the team that's specialized and works on this gene because that's what they've been working on. The problem is that the model organism researchers are submitting these genes, but they only submit genes that they're familiar with. And so typically that is about 4,000 genes in human, and that leaves 16,000 genes out of the picture. And those are the genes that we like to work on. So in a way, that's kind of complementary because we've been discovering a lot of new human disease by focusing on genes that were not known or were not studied. So this matchmaking efforts between all these different groups is really important. And we think that by continuing to do that, we'll continue to discover a lot of new human disease. So the challenges are that we're actually inundated by requests from all around the world. It's a good challenge, but we're getting submissions from all kinds of European countries, and recently very large collections of mutations from China, where gigantic pediatric hospitals have an incredible supply of patients. And so we're a bit overwhelmed by the demand. And I think this shows that we need to continue to do this. And we also need to continue to work with these model organisms and support them, because it's really clear that there's a lot of genes that remain to be identified in many of these species. And you can see that here. The next major issue is collaborations. We've become aware that there is a very different culture between the model organism researchers and the MDs. And that culture is hard to deal with. We at Baylor obviously have the model organism and the clinical portion of the research, and we participate and talk to each other, but the physicians are not inclined to explain what they do to the model organism researchers, and the model organism researchers are not easily inclined to explain to the MDs what they're doing. There's also a cultural gap, and when a physician approaches us, we know there's going to be 40 authors on the paper. In a model organism research community, this is never the case, and so it's a completely change of mind that needs to be brought about, and it's not easy to deal with. It seems like a lot of people in the model organism community are completely turned off by these issues. And so it's something we need to think about because this culture is a major hurdle. And so we're writing a white paper to explain to people how you got to deal with this, because it's clear that I get calls from people and saying, how do you deal with these issues? Because it's really not a simple issue. The other thing I think is important is that there is a language gap between the MDs and the MOs, the model organism researchers. And that language gap is not easy to bridge again. And we're trying to do this at Baylor between the model organism and groups and the human geneticists. But it can only be done by extensive interactions. And it's really been clear to us in the UDN that the more we talk to each other, the more we interact with the physicians, and the more phone calls we have, the better the projects get. And that's something that I think really should be emphasized. The other thing I already mentioned is that there are a lot of bioinformatics tools, but they're not transparent. They're not easy to use. And we're really trying to make that use so that everybody in an eye blink can look at a lot of data and get all the data interpretations and to integrate all these data in a simple program. Now, in the model organism community, there is an unappreciated element, and that is we can screen for drugs very quickly. By working out the pathways, we can screen for drugs quite rapidly. And this should be used more, we think. In fact, at the Neurological Research Institute where I am, we only work on drugs that pass the human cell test and the fly test, and that has been a successful approach. Now, there's challenging issues, and some of this will be brought up in the next talks, and that is, how do you study digenic, oligenic, and polygenic inheritance in model organisms? How do you deal with environmental gene interactions? And how do you deal with mutations that are not in the coding regions? Those are next to impossible to tackle in model organisms. We've tried, and that's a real challenge, and we'll hear more about that. Finally, I want to end with the benefits. It's been really gratifying to work with patients. I've never had so many emails from what I call tiger moms for their kids. As soon as they know that their kid has a mutation in a gene, many of them start organizations and group together, and they're actually some of our best supporters, and your supporters too at the NIH, and they're really eager to work with us. And a thing that's not emphasized enough, I think, in the rare disease research area is the fact that we dig deeper in these genes and have been able to make connections between some of these genes that from rare diseases with Parkinson's disease, amyotrophic lateral sclerosis, Alzheimer's disease, and have been able to work out pathways that connect these rare diseases to these late onset neurological diseases. And I think that's really an important path. Finally, we think that the pediatric disease, especially the very severe psychiatric cases, will be important for us to collect right now and start working on them to see if we can help with the discovery of genes that cause psychiatric disease. And we're doing that at a large scale with Texas Children's Hospital. Now, I'm going to end here and give credit to the people. The two teams for the Undiagnosed Disease Network, the zebrafish team and the fly team. The Baylor Comp team that has been very supportive and very helpful to work with us and make all these mouse knockouts. And the Ankle 2 team at UCSF. Thank you.
Thank you. We're going to take questions afterwards, so if you could please have a seat. And I'm now going to introduce Leah Starita to give her talk. Thank you.